Welcome to the Holistic Psychiatry Podcast. I'm Courtney Brown Snyder, a physician and holistic adult and child psychiatrist. In this episode, I'll be discussing how our microbiome influences our mental health. I'll be giving a sampling of some of the research studies that have looked at how particular microbes appear to impact particular brain conditions. Our gut microbiome is a thriving ecosystem consisting of trillions of microbes, bacteria, viruses, fungi, belonging to thousands of species, many of which were here before we were. Despite the emergence of research into the microbiome in the last 20 years, our ancestors recognized this link between digestion and emotions, mood, and behavior. Fecal transplants actually date back to ancient Greece and 4th century China. The gut-brain axis was already a thing by the 18th century. 19th century physicians appreciated the impact of the gastrointestinal tract on the mind. In the 20th century, however, we took a step backwards when scientists reversed this understanding, saying instead it was the mind that was influencing the gut. Finally, in the 21st century, it was realized that we can hold two truths at the same time. The brain influences the gut, largely via the autonomic nervous system, and the gut influences the brain through the microbiome. But how does the gut impact the brain beyond freeing up nutrients from our food? It appears to come down to three main mechanisms, each with endless complexity. First, microbes make products or soluble substances, such as neurotransmitters, hormones, and other metabolites that can cross the gut-blood barrier and thus move from the gut into the bloodstream. These substances can directly influence nerve cells. Some of these can cross the blood-brain barrier. They can also impact immune cells, which affect our nervous system. Number two, microbes can send signals via nerves of the enteric nervous system and the vagus, which connect the gut to the brain. The enteric nervous system is a mesh-like system of neurons of our GI tract that connects to our autonomic nervous system, which connects to the brain. Number three, microbes stimulate the immune system cells in the gut wall, communicating with the brain through cytokines, or inflammatory mediators or chemical messengers. In this way, they can be involved in neuroinflammation or brain inflammation. In short, some microbes make things that make us feel good and support our health, and some make things that make us feel bad and can contribute to illness. Research using mostly mouse models and some human studies examines what happens to brain symptoms when we increase or decrease certain microbial populations. The goal is to develop treatment interventions that target the microbiome. Psychobiotics, for example, would be probiotics, so microbial populations, aimed at treating mental health conditions. If you pull back, however, and consider the big picture, you'll realize that the goal for most of us is to increase microbial diversity, which has decreased due to our modern lifestyle. We don't necessarily need deep research to do that. So to start with brain development, the effects of the microbiome are already occurring as our brain is developing. The gut microbiome plays a role in the early development of neurons, which means they leave lasting impacts on the brain and thus potentially our behaviors. But what microbes was our brain being influenced by before we were born? Our mother's gut microbiome was making its metabolites that cross the placenta. One example is short-chain fatty acids. These impact the development of our brain cells. To give you a sense of some of the research into this gut-brain axis and what is being learned, I'll share a sampling of studies. Many brain-related conditions have been associated with changes in our microbiome, such as reduced amount of certain bacteria, especially those that produce short-chain fatty acids when they break down fiber. Butyrate is one example of a short-chain fatty acid believed to improve brain function. In a 2019 study, found a correlation between the amount of butyrate-producing bacteria and well-being. That doesn't mean we know if one is causing the other. Are the microbes providing a sense of well-being through their metabolites, or is a content brain and thus less stressful physiology a more welcoming environment for some microbes? Maybe it's both. Anxiety. Research shows that germ-free mice, so these are mice without a microbiome due to being reared in sterile conditions, are prone to anxiousness and are less sociable than those with an intact microbiome. Depression. The bacteria Lactobacillus rhamnosus appears to improve the mood of anxious and depressed mice. This doesn't happen, however, when the vagus is blocked, suggesting the signaling of the vagus is the mechanism of action. A 2019 randomized, double-blind, placebo-controlled trial, meaning the researchers and the participants didn't know who was getting the treatment and who was not. This study looked at the effects of a multi-species probiotic. Those in the probiotic group experienced a significant subjective improvement in mood compared to the placebo group. And I should say in the newsletter, each of these studies is linked to the original paper. Cognition. Another study found that giving probiotics to people with psychotic conditions, so these were individuals who had psychotic symptoms, the psychosis had resolved, but they were still having residual cognitive symptoms, and they found that giving probiotics improved cognitive function. Another study found that those who had used antibiotics for over 2 months scored lower on cognitive tests such as learning, working memory, and attentional tasks. Stress control. One small study with 45 participants showed that those on a diet high in fiber, prebiotics, and fermented food reported feeling less stressed than the control group. ADHD. Germ-free mice and those given antibiotics have also been found to be hyperactive, prone to risky behavior, and less able to learn or remember. Autism spectrum. We know that those with autism have greater rates of GI disorders. A 2013 study found that mice with a model version of autism had thinner and more permeable gut walls than normal mice. When analyzing the gut microbiome composition, researchers found abnormally low levels of a bacteria called Bacteroides fragilis and a 46 times higher 4-ethylphenyl sulfate in their blood relative to control mice. A very similar chemical, paracresol sulfate, is found at high levels in the blood of people with autism spectrum conditions. When researchers injected the non-autism mice with 4-ethylphenyl sulfate, it induced autism-like behaviors. Giving the mice with autism spectrum-like symptoms B. fragilis appeared to restore levels of 4-ethylphenyl sulfate to normal, improve their behavior and gastrointestinal symptoms. Social behavior. While research suggests that the microbiome contributes to autistic behavior, it also appears to contribute to the difference in social behavior in the general population. Numerous studies of autism-associated bacteria are also related to differences in social behavior in the general population. Aging and Alzheimer's. Aging mice have lower short-chain fatty acids, again, which are good for the brain, and they have dysregulated hyperactive microglial cells. These are inflammatory cells from the brain. Giving a high-fiber diet changed the types of bacteria in the gut microbiome, increased the production of short-chain fatty acids, and reduced the expression of certain genes that control inflammation in the brain. A follow-up study found that a high-fiber diet shifted most of the microglia out of a dysregulated state and back to the normal healthy state seen in younger adult mice. It is known that people with Alzheimer's have more inflammation-producing gut bacteria. Research shows that these changes occur even before the brain changes, which means it could be a target of intervention. As you can see, most of the research looks at bacteria. I wish fungi were also being considered, as many of us see candida and mold clinically wreaking havoc on the gut-brain axis. I've previously shared a post in the Substack newsletter on research showing evidence of candida and mold in the brains of those with Alzheimer's. Impact. The microbiome is difficult to study because of the differences in our baseline microbiome. Our early experiences can even impact that baseline. One study found that adults who have been formula-fed as children had a less diverse microbiome in adulthood. Children delivered by cesarean section have microbiomes that differ from those born vaginally. They also have a higher risk of early life infections, inflammatory conditions, and allergies. Our genetics, diet, social interactions, and exposures to toxins, EMF, and stress can impact our microbiome. Microbes, like the rest of us, can change when they feel threatened, in their case by multiplying and becoming more virulent. This has been studied in bacteria exposed to EMF. Increasing microbial diversity. Regardless of our differences, it seems that a diverse microbiome is important. I'd like to share ways to increase microbial diversity, but keep in mind that while this can be beneficial for most people, if someone's dealing with a high immune reactivity, food sensitivities, or having problems with particular food families, then some of this may not apply to them in terms of where they are in their healing process. But generally, the goal is to increase microbial diversity when possible. So a diet that's highly processed or high in carbs is going to lead to less microbial diversity. Prebiotic foods will increase microbial diversity, including things like garlic, onions, asparagus, cabbage, beans, and bananas. While fermented foods such as yogurt, kefir, sauerkraut, or other fermented vegetables can be helpful in increasing microbial diversity, many people I see, because of histamine intolerance, mast cell activation, or particular genetic variants such as DAOA or MAOA insufficiency, can have difficulty tolerating these very high histamine foods. Probiotics, as you can tell from the market, there's a plethora of options, which shows that we don't have it all figured out, but there does appear to be benefit. A more adventurous diet, for those, again, who can tolerate such adventures, even international travel has been shown to increase microbial diversity, possibly due to novel microbes and varied diets. Avoiding things that decrease the diversity of our microbiome, so again, sugar or high-carb diet, toxins, overuse of antibiotics, non-steroidal anti-inflammatory medications. Exercise can benefit the microbiome, but only if it's not to an extreme, which lowers diversity. Meditation or other practices that lower stress has been shown to be beneficial. Being socially active, which actually results in an exchange of microbes, which I hope to explore in a future newsletter and episode. Getting outside in the natural, unsanitized world. And because I'm a fan of zinc, I'll mention it here again. Zinc deficiency can alter the microbiome, and zinc supplementation has been shown to protect the gut microbiome from pathogens and toxins. Keep in mind, however, that too much zinc can have negative impacts on the microbiome, and it is one of those nutrients that we check levels and monitor accordingly. So I hope I've given you a deeper understanding of the wonders of the microbial world living within each of us and how we can support it so it can support us. If you'd like to receive the newsletter in your mailbox each week, please consider subscribing at CourtneySnyderMD.com. Until next time, take care.